Is this your desktop?
Yeah.
Okay. Right, here's the paper. So this paper is about thirst, right? 2 forms of thirst. It's a pretty straightforward paper, I thought. And maybe more interesting aspect is the technology on what they did. But they start off by talking about the 2 forms of thirst. One is when there's osmotic pressure. So if you inject it, so I think what they do is inject 2 molar sodium chloride into the peritoneum, so that there's suddenly a lot of salts in the peritoneum, and that the body will require a lot of water to counterbalance the amount of salts that's there. So that's the osmotic thirst. And then they talk about using furosemide, which is a diuretic. And if you inject that into animals, then the animal will pee a lot, but they'll be peeing a lot of salts with it. So you can even see crystals coming out of in their pee. It's that severe, right? So this is thirst or dehydration and a severe loss of salts in the body. So then the body tries to compensate for that by drinking liquids, but also having a hunger for salts. So it's one of those cases where, like, if you're salt deprived, then you want to eat something very salty. All right, so since Amanda's not here today, who's going to be doing figure 1?
I guess I can.
All right.
Okay, so in the first panel A, they had 4 different groups where it was control. Another group was under osmotic stress by injecting hypertonic solutes. And then there's the hypovolemic, where again it was injected with furosemide. And then lastly was the water deprived. And in panel A, they are looking at the different FOS expression across the SFO and the OVLT, which are the 2 neural, or the 2 areas that are involved in perceiving thirst. And so that's the different FOS expression. In B, they showed that when they injected the hypertonic solutes, the mice prefer to consume just pure water to lower the osmolarity back to normal levels, while for the hypovolemic group, they preferred, or they showed equal interest in both water and salt solutions, so they can, since they lost a lot of salt level, they try to go back into main, or like the normal
levels
range again. And water deprived animals, yes, thank you. Water deprived animals showed preference of water over salts, and sodium deprived consumed sodium more specifically. And then in C, it was just like they measured the blood volume and the osmolarity between the various groups to make sure that the effect with injecting the hypertonic solutes or the furosemide indeed had an effect in both the volume and osmolarity. And then for D, they wanted to see if the osmotic and hypovolemic stresses are detected by the same neurons. And so they employed an activity-dependent genetic labeling. And they noticed that when they injected hypertonic saline, it induced a lot of robust labeling in SFO and OVLT. But when they did a similar thing with hypovolemia group, there were significantly fewer cells that had labeling in the SFO and OVLT. So this suggested that the osmotic thirst and hypovolemic thirst activate two different sets of cells within the CVL.
Very good. Just a little bit more background with the SFO and the OVLT. Those are two regions that when, let's say, if we look at the neuronal activity in these regions in animals that become thirsty, as you can see here with CFOS, you can see that these neurons are activated when the animal should be feeling thirsty, both in osmotic and hypovolemic conditions. Also, if you were to stimulate these cells, then it can translate into drinking behavior. So if you were to use channel rhodopsin and use light to activate these neurons in the SFO and the OVLT, then the animals will, once you turn the light on, the animals will race towards the spout and start drinking, even though there's no reason or homeostatic reason to drink. So these are the two regions. And then a little bit of background on D, the trap mice. I think we've been talking about this over several papers, but this is a CFOS promoter. So this trap mouse has the CFOS gene and then a CRE-ER. So tamoxifen or OHT is a hydroxy form of tamoxifen. So it's the metabolized form of tamoxifen that can act instantly on the ER part. So once the ER part of the CRE-ER binds to OHT, the CRE-ER can translocate to the nucleus and recombine whatever is there. So in this case, they probably did trap with TD tomato or something to label the cells that were active during this process. I think it's interesting that they inject the tamoxifen, hydroxytamoxifen, an hour after their initial stimulus, right? So it probably happens pretty fast. They leave that animal for 6 hours and that's presumably the time it takes for the red labeling. And then they go back and give another stimulus, saline or sodium chloride or something else a week later. So the cells that were active with the initial stimulus, say the left-hand panel with saline, the cells that were activated by saline, which are not that many in this region, of course, would get labeled during those 6 hour period, presumably, right? They're not really measuring, they're not doing anything for the 6 hours. It's just, they're saying maybe the optimal labeling time is during those 6 hours. Same thing for the middle panel. So they inject sodium chloride into the peritoneum. There's osmotic stress on these animals. They inject the tamoxifen, during which time the cells or the neurons that are activated by this osmotic stress are labeled in red. They wait for everything to stabilize for a week. And then a week later, they inject the same 2 molar sodium chloride again, and they see that green cells and red cells are largely overlapping. There's some cells that are not red, but green, not green, but red. But there are also a lot of yellow cells, which are red and green. Right. So this is osmotic stress, and then 1 week later, looking at osmotic stress again, as opposed to the right-hand panel, which is osmotic labeling with red with osmotic stress, and then giving furosemide a week later and looking at hypervolemic neurons that are conveying hypovolemic information. And those cells, red and green, don't overlap that much. Okay. Any questions about this figure?
Yes, actually.
Yeah.
If they're waiting a week after injecting hydroxytamoxifen,
Mhm.
why use hydroxytamoxifen and not just tamoxifen? Because it takes about a week to.
Yeah. So tamoxifen is the cheaper version. But once tamoxifen gets into the body, it has to get that hydroxyl for it to bind to the ER, right? And that happens in the liver. So then the time that it takes tamoxifen to get metabolized, there will be a lag, a delay for that to happen, right? So then you might not really be able to capture the effect of sodium chloride.
Oh, okay.
I mean, it will still
sense.
happen, but maybe the efficiency will be lower because it'll kick in maybe 3 hours later, 4 hours later, 2 hours later, who knows? But it's not as direct as using the more expensive hydroxytamoxifen. Significantly more expensive, probably like 20 times more expensive, right?
And
Okay.
I have a question for Dee. How does the C-force expression pattern that you capture 1 hour later versus, let's say, oh, not 1 hour later, but yeah, 1 hour later versus from the beginning, let's say. What's the difference between those two? Are they actually captured both at the 1 hour time point, or are they different?
You're saying
why there's a delay, or what about, let's say, injecting the
tamoxifen before the saline?
not even before or even at the same time of injecting the saline?
Could be.
What's the reason behind that? Is that because of the?
I guess if you were to if we assume that hydroxytamoxifen is instantaneously working so that the Cree become, as soon as you inject hydroxytamoxifen, the Cree translocates into the nucleus and does the recombinating that moment, then you would be capturing the effect of NaCl 1 hour later, which is exactly what you're doing with CFOS, kind of. Right?
Okay.
I don't know if there's a good explanation for it, because also for the CFOS, you're waiting for NaCl to kick in, the neurons to get activated, to make the CFOS mRNA, and then that mRNA to translate into protein before you can capture it, right? So there's some time difference. But I don't know why they injected the tamoxifen later than the stimulus, but I think we can justify it.
And in B, how does furo was used for hypovolemic versus sodium deprivation? Did it work in both ways, or?
I figure that they must have supplied food that has sodium versus food without sodium,
Okay.
because they probably wait a couple hours after furosemide injection to do this.
So it's not a sole drug effect. It has to do with the feeding.
Yeah, it's not. Yeah.
Okay.
Yeah. Also, while I was reading this, I saw that they were using the cell signaling antibody from the
For
CFOS
CFOS?
antibody from cell signaling. Yeah.
Okay.
I'm thinking that we should get this. You know why? Because the CFOS antibody that they're using is a rabbit, right? And if we didn't have any reason to, let's say we want to, we're just staining for CFOS, it seems like this CFOS antibody is working pretty well. So we should just use this and save the GOAT antibody for when we need to do co-stainings. Because we'll never
That's
get
actually good. Yeah, that's actually a good point.
Yeah. So why don't we get the cell signaling antibody, use that for most things, and then
Yeah.
whenever we need to co-stain with a rabbit antibody, for example,
Yeah.
then we use the GOAT antibody. Yeah. So I'll leave it to Nancy to put in that order. Yeah. All right. So figure 2. It seems right that Yue goes over this figure.
Yeah. So, so in figure 2, they are doing a single cell RNA sequencing analysis on the brain tissues that are specific to the taste, that is the SFO and OVLT. So what they did basically is they physically separate these two regions and dissociate them and send them for the RNA sequencing.
Yeah.
Actually, I checked their
Then,
method.
Yue.
Yes.
Just one thing I forgot to mention, or I didn't emphasize, is here they're saying that when you look in this panel, that osmotic versus hypovolemic are going through different neurons, right? So they're trying to identify or find ways to characterize the subpopulations. How do you you want to find markers or genes that are enriched in the red population and genes that are enriched in the green population. So that's what they're trying to do, right? Okay.
Yes. And so they send it for sequencing, and then they use Seurat to analyze the data and come up with the UMAP distributions. So they're able to separate 12 to 13 groups on each sample. And most of them, they're able to assign them the cell type. That is what A and B stand for. And then in C and D, they are trying to characterize the neurons based on being excitatory or inhibitory. So overall, they're able to identify, I think there are 5 excitatory and 3 inhibitory neuron groups in SFO, whereas there are 6 excitatory and 2 inhibitory subgroups in the OVLT region. And then they try to, in E and F, they try to map out these excitatory neuron groups and find gene markers for them. So it's a very typical single cell RNA-seq analysis. And yeah, that's.
So here it's completely unbiased. They're just taking SFO, dissociating all those cells, and then not even picking out neurons. They're seeing astrocytes,
yes.
They're seeing fibroblasts, all sorts of cells that are there, right? And the same with OVLT. And then they I guess based on that information, they take this cluster, for example, in the SFO neurons, and then they subdivide that based on They take clusters within this cluster, and then they take clusters within the inhibitory neurons, and the same with the OVLT case. Trying to come up with subpopulations. Right? Okay, next figure. Do you just want to go do the next figure also, since it's pretty simple?
Oh,
Straight-
the next figure, I think they also applied 4 different conditions to the mice instead of like a totally. So in previous figure, they just didn't analyze the like a regular mice. So here, I think they applied 4 different conditions, and then, well, what they did, and then they want to see the reaction or like the response of different conditions in corresponding to the excitatory neurons that they identified previously. So here they are able to, so in B, we can see that when it's set it, the mice doesn't have like a response. So that's pretty normal. And but between the osmotic and hypovolemic conditions, there are distinct neurons that are expressing FOS, indicating that they are excited during these 2 conditions. So in C, I think they're just matching these different conditions in with the group of excitatory neurons they found, and they are able to conclude that the 2 conditions, osmotic and hypovolemic, will be activating different subsets of the neurons.
Does anyone remember if they were sequencing just the tissue as a whole, or were they selecting for neurons?
You mean when they send that for single cell sequencing?
Yeah.
In this case?
Yeah, in this case.
Maybe
it's the same.
I think it's the same method. They just dissociate everything. Are they able to select for neurons for sequencing only?
If they had a neuronal marker, but I can't remember if they did that.
Oh, okay.
I don't think they did.
No, it's just a bulk tissue.
I have a couple of questions.
Yeah.
When you run the cells through RNA-seq, how do you know which ones are excitatory versus inhibitory? Is there just like a group of genes that's like, oh, these are expressed by all excitatory neurons?
Mhm.
Is that how that works?
Yeah, I think they just go by glutamate versus GABA transporters.
And then when they are showing the FOS expression, they're saying that it's normalized. Is it normalized to the sated state? Like
Could
what?
be.
I'm just like kind of lost on what did they normalize the FOS expression to? I'm guessing to the sated state, but I'm not sure.
Mhm. It's a good point. I don't know what they did to normalize, but my guess is maybe they just bulk normalize and see what the average CFOS is in the neuronal population, for example. Because otherwise, if it was just a sated state, then if they were to take all the sated or all the neurons from sated state, would you still see CFOS there? I guess you could. Yeah, I don't know what the answer to that is, but so it seems. So the reason that I asked whether they came from neurons or from bulk is first to figure out if they did this experiment multiple times. So did they do figure 2 first and then go on to figure 3? Or did they just take sated from figure 2 and make that, or sated from figure 3 and use that as figure 2. But but the cell numbers were smaller here. So if I recollect correctly, they use fewer cells in this experiment somewhere. Anyways, regardless of what they did, I think give me 1 second. I don't know who this is. Hello? Okay. So they made a big point about coming up with this amazing strategy that they, as if it was some technical advance, by saying that they used. So when you're dissociating cells from a tissue, that's very stressful for the cell. So that will definitely activate some cellular processes that you might not want to see. Right? So Alex, when we're dissociating cells, we're probably also seeing that, where there are things that are caused just by that stress of dissociation.
For sure.
Yeah. So they said that and all they did actually was to add actinomycin D to the entire process, essentially. What does actinomycin D do? I feel like Jasmine might know this best. No?
You know, I don't know, but I'm willing to learn.
It's a transcriptional blocker.
Right.
Yeah. So it just blocks transcription. So as they kill the mouse, they take the tissue out, and then they start treating this tissue with actinomycin D, so that there's no new transcription happening during this entire process until they extract the cells and the RNA from cells. So anything that happened from the moment that the animal was dead to now is you can disregard or you won't see it by treating actinomycin D. So if you think about it, it's pretty logical to use actinomycin D. Okay, so they again, they just take animals from sated, osmotic, hypovolemic, and water deprived conditions. They take out SFO, they take out OVLT, they sequence single cells. I feel like it's thousands of single cells in each case. And then they look for CFOS expression in each cell type or cell, right? And they want to see if a certain cluster lights up more in a certain condition versus another condition. Yeah. And based on this, image, what would you guys concentrate on? Well, because it's pretty broad, right? I guess if it was up to me, maybe BMP3 population seems to be active, quite active in the hypovolemic condition in the OVLT, right? maybe this population here also. And here maybe this,
Yeah.
and the osmotic might pick this one or this cluster. So they focused in on RXFP1, right? In the next figure. But in any case, does everyone understand what they're trying to do here? Yeah. So based on this single cell analysis, single cell sequencing analysis, they find some candidates that might correspond to a specific condition, osmotic versus hypovolemic, right? And that leads us to figure 4. And who will explain figure 4 for us? Anybody? Nobody? Well, I went over most of it, right? When they're trying to find a genetic marker, a transcript that's enriched in the sated condition or in the osmotic condition versus the hypervolemic condition, right? And based from here, we try to pick out which ones might be good markers, like the BMP3 here, or the HTR7 here, or the RXFP1 here, LYPD6, right?
So here they are trying to find a marker that can approximately be consistent to the expression patterns in each region, representing either the osmotic or hypovolemic conditions. So that's why in A, they find that the RXFP1 looks pretty similar to the, or I think, is this a region specific or is it condition specific? Sorry.
I think it's both, because RXFP1 seems to be in both regions, right?
Yeah. Well, but what they're claiming here is that expression of RXFP1 and pyronorphin strongly correlate with under osmotic and hypovolemic thirst respectively. So I think this still kind of condition specific. Then they define RXFP1 to be a marker for osmotic condition, whereas pyronorphin function as a hypovolemic stress condition.
All right.
But well, I mean, if for example, if we look at the A top panel, the false expression of osmotic, the RXFP1 might be pretty similar as when there is like in the neuronal group 4 and 5, you also have overexpression of RXFP1, whereas in the OVLT region, it's not quite the case, right? I mean, you can see several groups. So that's the bottom panel of A. You can see several neuronal groups having false expression when experiencing osmotic condition, where you only have one of the groups to be strong correlation with RXFP1. So I don't know if, but I mean, they also claim this is just like an approximate marker. It's not like a definite marker for two conditions. But pyronorphin, you can see the same thing, like for false expression in SFO, you have pyronorphin expression when you have false in hypovolemic condition. In OVLT, it's probably kind of similar. So it's kind of hard for them to conclude a one strong marker for that represents all the neurons in each condition, but they did find two candidates. So that's why they are trying to do this overlap in B and C, and they found, oh, that pretty similar. Let's just try a manipulation to see if that works. So in D, they are trying to do the optogenetic manipulation to activate these neurons, and they find that when they are doing the light opto activation on the RXFP1 mice, they can, in both regions, they observe a significant increase in water consumption, but there's no increase in the salt solutions. So which is representing that it is consistent to the osmotic thirst. Whereas in the buprenorphine, which is on the right too, when they activate these two neurons, they can find both water and salt solution consumption increased dramatically. So that is representing the hypovolemic condition. So they're concluding here, these two markers do mostly represent the two conditions. I mean, or at least they play a role in these neurons expressing these two markers play a role in each condition respectively. And finally, in E, I think they are using a chemogenetic inhibition of these two markers, and what they are finding is pretty similar. I can't remember the exact step they did, but they're testing the legs, right? Yeah. So compared to the RXFP1, buprenorphine have a higher leg. I think they're saying like if they inhibit these neurons, they do observe a water like a consumption decrease. Is that true for this part?
Yeah.
Okay, strongly. So chemogenetic inhibition of RXFP1 positive but not buprenorphine positive neurons strongly suppress osmotic stress-induced water intake. Okay, so they do, I think they also preset the conditions. Overall, they are concluding the same thing. It's like RXFP1 is representing osmotic neurons, and then buprenorphine is representing the hypovolemic stress neurons. Yeah, that's basically what they're talking about. My question here is that if they are eventually going to find this unique markers for the neurons that are representing both conditions, what is the purpose of characterizing 5 distinct excitatory groups at the first place? They're not using these markers anyway because they couldn't find a correlation. So why don't they just separate them into 2 groups and then just find a marker for each?
So I think if we look at figure 1 here, right? And let's say you came up to me and said, look, there are 2 different populations of cells, of neurons, that seem to be one for osmotic, the other for hypovolemic. And I want to characterize these. I want to find markers for these, right? So then from that point, the strategy is pretty clear. You have to do single cell sequencing, characterize the sequences, or do sequence analysis, and then find markers for the clusters that are there, right? And then get mice, Cree mice for each of those clusters, and do the gain of function and loss of function experiments there, right? So right. I think by the time that you have this first figure, your roadmap is pretty clear on what you have to do. Yeah. So they do all this work, and then they do this, and then at this point, probably, if you were doing this project, you will come up to me and say, look, I need to make mice from Jackson that has HTR7 Cree, CalCR Cree, EFA5 Cree, right? And then But the other 7, we don't have Cree mice for at Jackson, so I need to make those mice because I don't know which one of these are going to really work, right? Which is probably what they did. I mean, the Oka lab now has a lot of money, so they probably made a lot of mice. They talk about making the RXFP1 Cree mouse. Buprenorphine is available in Jackson, right? So then they probably systematically went through each line that they have. Alex, you know how long it takes to make mice and then to characterize each one. Let's say you get like 5 founders for each one, right? Comparing each of those 5 founders doing the experiment. And largely, I think they probably tested BMP3 Cree, didn't see anything interesting, right? So that's the danger of doing these fishing expeditions. We don't know if the clusters, even though here, look, based on this, I would have definitely gone on for BMP3, and they're not showing BMP3. So that probably means that they tried out BMP3, didn't work, right? Based on this, I don't know if I would have settled on RXFP1, right? It seems like LYPD6 might be a more discrete population. And part of this is because all this clustering is arbitrary. There are algorithms based behind it, but it doesn't necessarily correlate with function. Right? And this is based on statistics. Sometimes biology is qualitative, meaning that maybe you don't make tons of a certain gene, you just make a couple copies, and that's what matters. Right? And what we have to remember here is these markers, say HTR7, is just a marker for that cell. HTR7 in itself might not have anything to do with this process. Right? So that's why there's a disconnect between function and the gene. Right? If we were looking specifically for, let's say we do this in the taste system and you want to find what the sour receptor is, right? You can make clusters based on different transmembrane proteins. Maybe that'll make more sense. And then you go after those transmembrane proteins. Here, it's all sorts of things. I don't even know what RXFP1 is. Does anyone know what RFXP1 is? But there are all sorts of things here. There are growth factors. There are axon guidance molecules, random genes. So this is purely based on statistical analysis of which genes are expressed in certain clusters, but not in others. Is anyone Googling RXFP1?
I think it's a relaxing insulin-like family peptide 1 receptor.
What the heck is that? Right?
So for those genes, there is like a relaxin is a protein hormone.
Mhm.
And it's basically critical in the reproductive process,
Mhm.
sperm mortality, pregnancy, parturition. So mostly, who will think that will remotely relate to osmotic stress?
Yeah,
It's like-
again, the point is that the function of these genes have nothing to do with the cluster. In most cases, right?
Yeah.
In most cases, the genes that pop up are not and don't have any relevance to the function of that cluster. And that's based on the assumption that the clusters are meaningful to begin with. We don't know if the clusters here are meaningful. You did this during the summer when you were clustering, right? You just have to change probably one parameter. It can go
Oh, you
from
can
12
get,
clusters.
if you want
You go from like 12 clusters to 3 clusters, right?
yes.
I mean, I think these clusters, if you look in figure 2 of, figure 2, these clusters are meaningful, right? Because these
Yes.
correspond to exons versus non-exons, excitatory neurons versus, what did I say exons? I meant neurons versus non-neurons. But you can divide them up into excitatory neurons versus inhibitory neurons. But these are based on very well-known cell types, right? You would expect fibroblasts to be different from astrocytes, for example. And there's probably some kind of a weighing that goes on in to weigh some marker genes that are specific for neurons versus non-neurons. They probably have that kind of a filtering to begin with. Because I think if we did this blindly, they probably won't come up with clusters like this. Let's say you take skin cells, right, and you do this, then maybe the biggest driver of the different cell populations is what cell cycle what stage of the cell cycle that cell is in, right? And you make clusters, you might end up with 3 based on the cell cycle. So there are many gene programs that are running at the same time, and they must have, and when they did this analysis here for figure 2, they probably had a filtering system to group the neurons together, the fibroblasts together. But basically what I'm trying to convey is that that this is still quite arbitrary, because there are 20,000 genes that you have to look through. You don't know if you have, well, you're never going to have complete information, because just single cell sequencing isn't sensitive enough. So you're only going to capture genes that are relatively highly expressed. So by the time you get here, you have to make the decision on which mice to make and what to go after. And to me, this from figure 3 to figure 4 is very underwhelming. You can see that they probably didn't get good results from their favorite candidates. Right? But all in all, they find that RXFP1 shows might be good markers for osmotic versus prodynorphin. I didn't look at the extended figures, but do they show that prodynorphin and RXFP1 are non-overlapping cells in situs?
Should be.
Should. Or
So-
this is meaningless.
So for example, if they were to do this kind of experiment, right, then they would be able to show that these red cells are RFXP1 expressing cells, but not prodynorphin neurons, right? Vice versa.
So basically, what does the RNA-seq data doing in this paper?
RNA-seq data gave you RXFP1. I'm sure they looked at prodynorphin before, because they had prodynorphin papers before, right?
Yeah.
So after all this work, they found RXFP1.
And
But-
prodynorphin doesn't really pop up in the RNA-seq clustering.
No.
And also they claim they have a new method. It's not a new method.
Mhm.
they put a fancy name like stimulus, RNA, whatever. My opinion is
Yeah.
Anyway,
But if they are able to keep the animal, let's say,
We can't hear you.
hello?
oh, sorry, I need to mute.
I'm just wondering if they are able to apply the animals with continuous stress condition. Is it applicable to the taste as well? So we keep the mice like getting continuous stimulus as one taste, and then we send their brain for RNA sequencing, and we find if that will be corresponding to a certain group of like a certain neurons will be activated, and then we find a marker on that. Is it possible here?
I missed the first part, so
Oh, I just wonder if it is possible we can do similar studies as figure 3. We apply a continuous stimulus to the mice, and then we send their brain for RNA sequencing and compare it to the normal ones. Is it possible, and are we going to find something from there?
yeah, actually, so let's say prodynorphin, no, not prodynorphin, P2X2, GQ, right? You could do that, right? So that might be worth thinking about, Yue. You know, where you do a GQ experiment, just sequence with actinomycin D, and then sequence the CNO plus minus CNO, right? Or plus minus GQ. Yeah.
Yeah.
I mean, I don't know if you really need to do the minus GQ even. You can just sequence and see what markers you get. I would think it's more informative to get more cell numbers if you were to do just the GQ CNO. So activate the taste system using the P2X2 GQ CNO. And if you could, let's say the total number of cells you can sequence is 10,000, right? Wouldn't you rather take 10,000 of neurons from that mouse rather than 5,000 of the stimulated and 5,000 of unstimulated? I think that would be much more informative.
Right. And they are just combining all samples from all the mice they are doing experiments on. So that means if we do enough mice, we are able to collect as many cells as possible as we want. It's also one way to do it. Yeah.
I mean, there are multiple ways to do this. Perhaps we should think about this, and you can sequence from the P2X2 animals.
Yeah.
You can get your figure 2. You know? Anyways, think about it and we can talk. We're 1 minute over the hour. So.
Yes.
Okay.
All right.
No more questions then, right? No, we're good. Okay. All right. Thanks, everyone.